Hello everyone, welcome to Lab 8. Today we're gonna be discussing about muscle. So first let's have some summary questions from last lab. Name the layer between the arrows. So this is the transverse section of peripheral nerve. So peripheral nerve externally is covered with epineurium, and groups of axons are covered with perineurium, and a single nerve fiber is covered with endoneurium. So the arrow points into the perineurium. Question number 2, what is this a section of? So obviously here we have a collection of neuronal cell bodies, so it's a ganglia, and there are three types of ganglia. We have dorsal root ganglia, sympathetic and parasympathetic ganglia, but this one is a dorsal root ganglia. The cell bodies are round in shape, the nuclei is found at the center, and the satellite cells form a complete ring. So this is a dorsal root ganglia. So in today's lab session, we'll try to see the three types of muscle, skeletal, smooth, and cardiac muscle. We'll see the features of these three types of muscles in longitudinal and transverse sections. So first, let's have an overview of the three types of muscles. So skeletal muscles, as their name indicates, they are found attached to the skeleton. And each skeletal muscle fiber is formed by the fusion of several myoblasts. So due to that, each skeletal muscle fiber is multinucleated. And the nucleus lies peripherally. It's pushed to the periphery. And in a longitudinal section, skeletal muscles, they have a dark and light band, which is known as striation. So skeletal muscles are striated. The second type of muscles are smooth muscles. They are found in the walls of hollow viscera. And smooth muscles, they don't have striations. They are non-striated. And they are mononucleated. They contain a single nucleus, and the nucleus is found at the center. The third type is cardiac muscle. Cardiac muscles are found in the wall of heart, in the myocardium of the heart, in the middle wall of the heart. And cardiac muscles, like skeletal muscles, they are striated. They show light and dark bands under the microscope. But like smooth muscle, they contain a single nucleus, and the nucleus is found at the center. And the other distinguishing feature of cardiac muscles is they contain intercalated disc. Adjacent cardiac muscle cells are attached by, or they communicate by, intercalated disc. So these dark lines that run transversely, they indicate intercalated discs. So now let us see skeletal muscle in detail. So first let us see the connective tissue coverings of skeletal muscle. Like peripheral nerve, skeletal muscle is also covered by three connective tissue layers. So most externally, the whole muscle is covered with a dense irregular connective tissue, which is known as epimysium. Groups of muscle fibers or fascicles, they are covered with perimysium, another dense connective tissue layer. And a single muscle fiber is covered with a loose connective tissue, which is known as endomysium. They are homologous with the three layers of peripheral nerve. We have epineurium, perineurium, and endoneurium. Here in the skeletal muscle, we have epimysium, perimysium, and endomysium. Now let us see sarcomeres. Both skeletal and cardiac muscles, they contain sarcomeres. Sarcomeres are functional units both in skeletal and cardiac muscles. So when we see these sarcomeres, they contain dark and light bands. So the darker band is known as A band. And A band contains myosin and actin. It contains thick and thin myofilaments. So it appears darker under the microscope and is known as A band. At the center of the A band, we have a relatively lighter area which contains only thick myofilaments or myosin, and this area is known as H zone. At the center of H zone, we have a structural protein that holds the myosin together. This structural protein forms a line which is known as M line. The lighter bands are known as I band, and I band is formed by only thin myofilaments or actin. And at the center of the I band, we have another structural protein that holds the actin myofilaments together. And it forms a line, and this line is known as Z line. So sarcomeres are the area between two successive Z lines. So this is one sarcomere. So during contraction, there are changes in these bands. For example, during contraction, the actin, it slides over the myosin and it moves to the center. So during contraction, the H zone, it becomes shorter or it may disappear. But the A band, it remains the same. Similarly, the I band also shortens. So the actin myofilaments, they slide over the myosin, so the I band also shortens during contraction. And during contraction, the Z line, they become more closer together. And when you see skeletal muscle in a cross section, skeletal muscles are polygonal in shape. And the nuclei is found at the periphery. And in a cross section, skeletal muscle, the cytoplasm shows stippling. It's not homogeneous or it's not smooth. It has a stippling appearance. This stippling appearance is due to the section of myofibrils. And in a longitudinal section, we can see the striations, these dark and light bands. And we can also see the peripherally located nuclei. Now let us see some slides from the slide box. So first let us see a transverse section of skeletal muscle. So when you see the connective tissue coverings of skeletal muscle, here externally we have a dense irregular connective tissue, and this is the epimysium. And here we have fascicles. These are fascicles, groups of muscle fibers, and fascicles are surrounded by perimysium. So this is one fascicle, so it's surrounded by perimysium. So this is the perimysium, which surrounds fascicles. And each muscle fiber is surrounded by endomysium. So this is an endomysium. It's a loose connective tissue. And when we see it with a higher magnification, the cytoplasm has a stippling appearance. Not only skeletal muscles, cardiac muscles has also this stippling appearance. Both of them are striated. So in a cross section or in a transverse section, both skeletal and cardiac muscle cells, they have a stippling appearance. Their cytoplasm has a stippling appearance. We have also other slides. Here we have a longitudinal slide section. So you can see the light and dark bands or the striations, and you can also see the peripherally located nuclei. We have also another longitudinal section with a different staining. So here you can see it we have this darker band, which is the A band, and it's formed by both actin and myosin. And on each side of this dark band, we have lighter bands, which are known as the I band, and they are formed by thin myofilaments or actin. And at the center of the I band, as you see it here, we have this dark line. This dark line is Z line or dead disk. And the region between these two successive dead discs, this is one sarcomere. And we have also a tongue section. Tongue consists of skeletal muscles mainly. And these skeletal muscles are arranged in 3 dimensions. So we can find both transverse and longitudinal sections in a tongue section. So here we have a transverse section with the stippling of cytoplasm and with the peripherally located nuclei. And here there is a longitudinal section. Let us see smooth muscle. Here we have both a longitudinal and transverse section of a smooth muscle. So as you see it in the longitudinal section, there is no striations. And the nuclei appears lighter in staining. The nuclei contains loss of euchromatin, so due to that it appears lighter. And as you know it from lecture, smooth muscle, it contracts from every direction. So when the muscle contracts, the nuclei becomes wiggly, or it looks like a worm. Here in a transverse section, you can also see the nuclei, and the nuclei is found at the center. And the cytoplasm doesn't have a stippling appearance, it's homogeneous because smooth muscle is not striated. So now let us see some slides from our slide pack. So first let us see small intestine. Small intestine contains both transverse and longitudinal sections of smooth muscle. In the outer part, in the muscularis externa of small intestine, we have these two sections of smooth muscle. Here we have a longitudinal section, and here there is a transverse section. So you can see the lighter staining nuclei, and here there is a transverse section. If we zoom in more in the transverse section, we cannot see the stippling. The cytoplasm is homogeneous because it's not striated. And here the smooth muscle is in a relaxing state. That's why the nuclei are straight. We have also a section from gallbladder. Here in gallbladder, the two layers are not clearly demarcated, but here we have a longitudinal section, and here there is a transverse section. And here clearly we can see the cell border, and we can see the centrally located nucleus and the cytoplasm. And here some of the nuclei are wiggly, or they look like a worm. So this muscle is in a contraction state. That's why the nuclei are wiggly. The last type of muscle is cardiac muscle. This is the cross section of cardiac muscle. We can see the stippling, and here it's a longitudinal section, and in a longitudinal section we can see these dark lines. These dark lines are intercalated discs. And in a longitudinal section, it's not shown in this picture, but in a longitudinal section we can see the striations of cardiac muscles. And when you see a slide from our slide box, we have a slide having both transverse and longitudinal section. So first let's see the transverse section. So here it's mononucleated, it contains one nucleus, and the nucleus is found at the center. And as you see, the cytoplasm has a stippling appearance because cardiac muscle cells are also striated like skeletal. Let's zoom out from here and see the longitudinal section. Here we can also see that the nucleus is found at the center, and cardiac muscle cells are branching. They give branches, and the branches will merge with adjacent muscle cells. Let's look for intercalated disc. So running transversely, these dark lines indicate intercalated disc. You can also see it here. You can also see it here. And we can also see the striations. That's all about today's lab session. We have covered these slides. I encourage you to study these slides and practice identifying structures. And good luck on your assignment. See you next time.